Good evening, everyone. I am your biochemistry mentor, Dr. Pooja, and today I will be discussing the FMG recall of the questions that were asked in December 2020 exam. Now, see, all of you must have, prepared hard for this exam. And if I have to rate this exam, or if I compare this exam with the questions which were asked, in your August exam, this paper was very easy, isn't it? I have also, seen the questions. The questions were very easy, and you could have solved them easily also, because there were so many controversies regarding that August exam. You people were being tortured also a lot for this August exam. But yes, this year question paper, this month question paper were very, very easy. So today we are going to, discuss the questions which were asked in your exam. For those who got them all correct, obviously congratulations to you in advance. And for those who were not able to crack this time also, don't worry, there's always a hope. So let's start up with the discussion of the questions. Now see, question number 1. Complex 4 of ETC is inhibited by. Now first of all, you should know ETC stands for electron transport chain. Right. And basically, as it is clear from the word itself, it is the chain of transport of electrons. Now, what is the use of this? These electrons combine with protons in the presence of oxygen, forming metabolic water. Right. So we can say that metabolic water is produced during this electron transport chain, because this metabolic water is actually a result of metabolism of carbohydrates, of fats and proteins. Right. Now the question is that it is inhibited by. So we are actually going to concentrate on the inhibitors of ETC. Now all of you are familiar with the 4 type of complexes of electron transport chain, and the 5th complex is of oxidative phosphorylation. So ETC plus oxidative phosphorylation is known as actually biological oxidation. So if we concentrate on electron transport chain, we should say that it consists of 4 complexes. Complex 1, complex 2, right? And these combine and they give their electrons to ubiquinone, which is coenzyme Q also. So you can say that ubiquinone and coenzyme Q is the common acceptor which is accepting electrons from complex 1 and complex 2. This is again a very important point about this. Now this complex 1 is simple NADH coenzyme Q reductase, isn't it? And if we talk about complex 2 is simple succinate coenzyme Q reductase. See, by one chart we can answer so many questions. So when these electrons are handed over this ubiquinone, which is a common acceptor, it handovers the electrons to complex 3. And this complex 3 in the end gives electrons to complex 4, which is actually accepting the electrons in the end to generate this metabolic water. Right. So this complex is cytochrome B, C1, and here it is cytochrome C. Okay, now here the question is what they are asking about the inhibitors. So if we have to name the inhibitors, if we talk about the inhibitors of complex 1, they are, I'll tell you a very simple mnemonic for them so that you can remember, because this is a very important question which can be asked. later. PCR. PCR, all of you know, it is polymerase chain reaction. You have to put A along with this. So this P stands for pyridine. C stands for chlorpromazone. This R stands for rotenone. And this N, A stands for amobarbital, which is a barbiturate also. So these are what? These are the simple inhibitors of, you can say, complex 1. Then if we talk about complex 2, I'll write negative here, that means inhibitors. If we talk about complex 2, it has 2 inhibitors, carboxyl and malonate. All of you know malonate is a, you can say, competitive inhibitor of succinate also. If we talk about complex 3 has BAP. This is again a mnemonic for this. This is BAL, British antileucide, timicin, and this P stands for fenformin, or you can say all the hypoglycemic drugs will be the inhibitors of complex 3. And last but not the least, complex 4 is cyanide, very important, H2S azide. So these are what? These are the inhibitors of electron transport chain, and that is what is asked in the question. Now if you know this chart, you have to remember. Now the students who have already cleared this, who clear this exam, so obviously they don't have to remember this chart, but yes, by some other means, if they are not able to crack the exam this year, please remember this chart. This is very, very important. Okay, now let's go to see the question. What the question says. Simple question is asking complex 4 of ETC is inhibited by. So obviously the answer is cyanide. So here the answer will be cyanide. Since we are discussing the inhibitor of complex 4, let's see what others are. Oligomycin is an inhibitor of oxidative phosphorylation. Malonate is an inhibitor of complex 2. Rotenone is a inhibitor of complex 1. So here it's clear the answer would be cyanide. And here I will just give you a brief introduction of uncouplers also. Inhibitors are different, uncouplers are different. As I told you that if we see this slide, ETC and oxidative phosphorylation are coupled to form biological oxidation, right? If an uncoupler is there, it will uncouple the two processes. So whatever energy is required for the transport of electrons, that will be dissipated in the form of heat. That is what is thermogenesis. Right, so I hope this must be clear to you, and this is a very simple question which was asked in the exam. Now let's move on to the second question. This question, same question I have discussed in my previous videos also. In fact, in every video of mine, I had shared this question with you because this is a very important aspect of, metabolism of phenylalanine. Let's see the question. A 40-year-old man presented with black pigmentation of pinna and sclera of eyes. The color of urine changes to black on exposure, on long standing. So what is the diagnosis? So many times I have discussed this question. Now, again, before taking up with the discussion of this question, I would just like to brief you certain things, as this is again a very important concept. In a sense that it is simple L-captonuria. Why not this? Why not this? Why not this? Because here the key word is black pigmentation of pinna, typical case of ochronosis, plus the color of urine changes to black on exposure to air. If you guys have any problem regarding any, disease, any enzyme, don't hesitate to ask. Since it is a live session, so it's a kind of interactive session. So whatever queries are there in your mind regarding this metabolism, you can ask me. Okay. So When we are discussing this, I will just discuss the, L-captonuria. Basically, this is a disorder of phenylalanine. And all of you know that homogentisate is converted to melyl acetoacetate, isn't it? Acetoacetate. And here simple oxidation is there. The enzyme is homogentisate oxidase. Now, since if this enzyme is absent, there will be accumulation of this homogentisate in urine. And this homogentisate will be then converted into quinone acetate type of substances. Or they are known as L-capton type of substances. And that is why this disorder is known as L-captonuria. And that is what is exactly asked in the question. And what are the typical, biochemical manifestations of L-captonuria? Hyperpigmentation is there. Leading to ochronosis. Right. If we see the pinna, there is, deposition in the intervertebral disc also. And a very important diagnostic feature of this is, which you people must be knowing also, that it gives Benedict's test positive. A very important feature. Benedict's test is positive here. And all of you know that if Benedict's test is positive, that means some reducing property is there. And that is what exactly happens. Homogentisate is itself a reducing agent. Second important point about this is that ferric chloride test is also positive here. So these are some of the features of L-captonuria. Basically, it is formation of L-capton type of substance where urine turns black on exposure to air. And you can see the image here. This is how this will be the color of the urine. You can see. Because there is oxidation of homogentisate. And you can see it can lead to arthritis also. And you can see the pinna. Right? This is a typical case of ochronosis. So the answer to this question would be simple L-captonuria. Now, since we are discussing this, other options also you should know. Phenylketonuria, as the name suggests, it is excretion of phenylalanine in urine. And the deficient enzyme here is simple phenylalanine hydroxylase. The enzyme which converts phenylalanine into tyrosine. Okay. Then maple syrup urine disease, it is again maple syrup urine. The urine starts giving the smell of burnt sugar. And this is actually due to the disorder related to branched chain amino acids. And mnemonic to learn branched chain amino acids is LIV. Leucine, isoleucine, valine. And then if you look at Hartnupp's disease, actually this Hartnupp's disease is named after the discoverer, who was, a Britisher, and it is actually due to defective tubular reabsorption of tryptophan. Okay, so these are the disorders, but here always remember, black color means formation of L-capton type of substances. So I hope it is clear to you, and the option, your answer to this is A. Moving on to the next question. Reverse transcriptase is used in. Reverse is ulta, opposite. And transcriptase. Transcriptase is actually a process which is transcription. transcription. And all of you know there is a central dogma of information, isn't it? Now, according to that central dogma of information, DNA is converted into RNA by a process known as transcription, right? And then RNA is converted into, RNA translates the information in the form of proteins. Translation. Right. Now here what is happening, and DNA is converted into DNA by a process known as replication. Now what the question says here? The question is reverse transcriptase. Reverse means it is opposite of transcription, isn't it? So if we talk about reverse transcriptase, it would be like this. That means we are running on in an opposite direction. That means we are forming DNA from RNA. Now, very important aspect about this is, or I will just write it here, this reverse transcription is a process of formation of DNA from RNA. Now, the DNA so produced from RNA will be known as cDNA. C stands for complementary DNA. Complementary DNA, and its sequence will be altogether different from the DNA which is produced. Right. So overall, if we see, let's look at the options first. The question says a reverse transcriptase is used in. So obviously it has to be the amplification of RNA. Now, if you look at the other options, DNA amplification means we are forming DNA from DNA. Here we are projecting RNA so that it can form DNA. Now, you must have been confused in seeing the third option, isn't it? Now, if you look at this option, option says DNA dependent DNA polymerase. No. If we have to name reverse transcriptase, it is dependent on RNA because we are synthesizing DNA, but the sequence is dependent on RNA. So it is dependent on RNA, but forming DNA. Right? And fourth is RNA polymerase. Now, RNA polymerase, polymerization of RNA. So this is an enzyme of transcription. Is it clear? So it will be simple A. Right? So reverse transcriptase is actually reverse of the process in which complementary DNA is being produced from RNA. I hope this question is also clear to all of you. After this, we come over to another important question on centrifugation. LDH gets deposited in. Now see, centrifugation is a process of, isolating the organelles. Centrifuge, we are applying the centrifugal force so that, there is fractionation, there is separation of the organelles. So this is a very important process in which we are actually, checking the purity of the organelles. Purity of the organelles. So obviously, if we are checking the purity of the organelle, that enzyme will be deposited in the organelle when we are centrifugating it. When we are putting in a centrifugal tube and we are allowing it to rotate, whatever enzyme concentration is there in that organelle, that will come. For this, obviously, in other words, I should say that it should be a marker enzyme of that organelle, isn't it? Means you are getting my point. Suppose we have been given a solution, in which different, cells are there, and we have to centrifugate it just to see which particular organelle is having that particular concentration of the enzyme. So we are looking for the purity, whether it is fractionation, whether it is centrifugation. So we will say that there has to be a particular marker enzyme for that organelle. So here the question is on centrifugation LDH. Now all of you know about LDH. LDH stands for lactate dehydrogenase. And this lactate dehydrogenase converts pyruvic acid to lactic acid. And this LDH enzyme is a marker enzyme of cytoplasm. Very, very important. LDH is a marker enzyme of cytoplasm. So obviously, if we centrifugate, it will be deposited in that particular organelle where it is present in bulk. And one very important feature about LDH is that LDH is an isoenzyme which exists in 5 forms. All of you must be familiar with this. LDH1, I'll write it here. LDH1, 2, 3, 4, and 5. And you know what? This LDH is actually a very interesting question for the examiners also. Somewhere or the other, this time they have asked about the centrifugation. Another important question is there on LDH, that which of the fraction of LDH rises in MI? Which of the fraction of LDH rises in liver diseases? So LDH is a very favorite question. Now, if we talk about other organelles, this mitochondria plays a very important role. Mitochondria being the powerhouse of the cell or a seat of respiration, marker enzyme for mitochondria is SDH. SDH stands for succinate dehydrogenase. Dehydrogenation of succinic acid. That means succinic acid will be converted to fumaric acid. And one very important feature about this enzyme is that this is the only enzyme which is present on the inner mitochondrial membrane. This is the only enzyme which is present on the inner mitochondrial membrane of TCA. Otherwise, all the enzymes of TCA are there in the matrix. And that is why it has a very low redox potential. These are very, very important points about this. Less redox potential. Because these are the type of questions which are asked in actually the exam. Because this year we were expecting, in fact, in this month we were expecting that questions will be more of clinical based, but only one question was, asked on clinic, biochemical manifestations. Otherwise, all were very, very simple questions. But at least we didn't expect such type of easy questions can be asked, and very simple and basic questions. So the marker enzyme of mitochondria is succinate dehydrogenase. Let's see other organelles. Ribosomes. Ribosomes, they are the protein synthesizing machineries. So obviously on centrifugation, they have a different concept. Ki sedimentation coefficient will be there. But if we talk about enzymes, enzyme related to translation will be the marker enzyme of ribosomes, which is simple amino acyl tRNA synthetase. Nucleus mein replication is occurring, transcription is occurring. So these are the processes which are occurring. So obviously the enzymes related to that will be the marker enzymes. But here the question is the LDH on centrifugation, it will be deposited in the cytoplasm. So the answer to this question is C. I hope this question is clear to all of you. Since again, I'm telling you, this is a live session. If you guys have any problem, don't hesitate to ask. Because this is the pattern of the questions. So obviously, those students are really lucky enough, if they crack the exam this time. But even if the students who somehow cannot clear this exam, don't get, disheartened. There is always a brighter day. So you can ask me any problem, any query related to these questions. Good evening, Mr. Khan. How did your paper go? Because these questions were very good evening, Mr. Khan. If you have any queries, again, I'm telling you, don't hesitate, just ask me. I told you, if we talk about centrifugation, all the organelles will be deposited, which will give the purity of the organelle. Chalo, now we'll move on to the next question. Enzyme deficient in von Gierke's disease. Now see, this question, when I was taking my video sessions with you, I had discussed these glycogen storage disorders. Glycogen storage disorders so many times. And we had discussed so many, clinical based questions on this. But I'm surprised no clinical based question was asked in this. Straightforward question is there, enzyme deficient in von Gierke's disease. So there is no second doubt also about it ki you students didn't answer this question. But still, yes, very good, Mr. Khan, it's A. So if we see, before taking up with this, let me just brief you about glycogen storage disorders. I'm not going to take it in too much detail because we have already discussed it so many times. But yes, this is a very important question and the aspects are very, very important. So if we talk about von Gierke's disease, this is type 1. Right? And this is actually type 1A. Type 1B is due to the translocase, I'll just write it, endoplasmic reticulum translocator or translocase. But if we talk about von Gierke's disease, it is simple deficiency of glucose 6-phosphatase. Okay, so let's quickly revise. There are actually, I'll tell you a mnemonic also to revise this. Von Gierke's disease, वो फिजिक्स केमिस्ट्री और मैथ्स में होशियार था. Okay, V stands for von Gierke's, P stands for Pom's disease, C stands for Cory's disease, A stands for Anderson's disease, M stands for McArdle's, H stands for Her's disease, and T stands for Tharouy's disease. So these are the basically type 1, 2, 3, 4, 5, 6, and 7. Right. And if we talk about the enzymes, von Gierke's is due to the deficiency of glucose 6-phosphatase. Is it clear? Pom's disease, it is a lysosomal storage disorder, obviously glycogen storage. That means if a particular enzyme is deficient, that glycogen will be stored in the particular organ. And that is what exactly happens here. Pom's disease, the enzyme deficient is acid maltase. In Cory's, it is debranching. In Anderson's, it is branching. In McArdle's, it is muscle phosphorylase. In Her's disease, it is liver phosphorylase. And in Tharouy's disease, it is phosphofructokinase. So overall, out of these 7 disorders, PMT means Pom's disease, McArdle's disease, and Tharouy's disease, they are muscle related, muscle storage disorders. Right? And rest of them are liver storage disorders. So obviously there is accumulation of glycogen in the liver, there is accumulation of glycogen in the muscle. Out of these, this von Gierke's disease is considered to be the common one. And if we look at the hallmarks of this von Gierke's disease, again a mnemonic is there, H3L. H3L stands for hyperuricemia, hypertriglyceridemia, and the third one is hypoglycemia. And L stands for lactic acidosis. Right? POMS disease, the basic character is, you can say there will be cardiomyopathy in this. For Cory's and Anderson's disease, the mnemonic is A, B, C, D. A stands for what? Anderson's disease. So B is branching enzyme. So Anderson's disease is due to the branching enzyme, and C stands for Cory's, which is due to the deficiency of debranching enzyme. Since debranching enzyme is there, so this is also known as limit dextrinosis. I hope it is clear to all of you. Then McArdle's, it is muscle phosphorylase. Right? Myoglobinuria is a very important biochemical manifestation of this. Right? So here the question is simple. If you look at this slide again, enzyme deficient in von Gierke's disease is glucose 6-phosphatase. And I'll give you a very interesting representation. If glucose 6-phosphatase is absent, this reaction won't occur. So there will be increased concentration of glucose 6-phosphate, will be diverted to form TGs, which will be diverted to form more of lactic acid and more of uric acid. Right? Branching enzyme, I told you A, B, C. Branching enzyme deficient in Anderson's. Acid maltase in POMS. And if we talk about glycogen phosphorylase is the rate limiting enzyme of glycogenolysis. Can anybody tell me, out of all the students who are attending my class, what is the rate limiting enzyme of glycogenesis? A rate limiting enzyme of glycogenesis. Mr. Khan, can you tell me the answer to this? Rate limiting enzyme of glycogenesis? It is glycogen synthase. Okay. So this was another simple question which was asked in the exam. I hope the explanation is clear to all of you. And I hope you people, if you got it correct, then you got 1 mark for this question. Moving on to the next question. Patient present with drawing of skin and appears as shown below. This is a one, picture question. Identify the vitamin deficiency. Anyone? What answer you people gave for this? It's a very simple question again. Here you can see whatever area is exposed, right? There is again pigmentation. You can see. And here you can see the pigmentation is around the necklace base area or neck. So this type of characteristic neck is known as cassel necklace. Okay, Mr. Khan, it's yes, very good. It's B, correct. Deficiency of niacin. And you know, one very interesting feature about niacin is this niacin is actually vitamin B3, which is synthesized from tryptophan. 60 milligram of tryptophan is required to synthesize 1 milligram of niacin. So if there is deficiency of niacin, it will lead to 3 Ds. These 3 Ds include dermatitis, which has already been asked in the exam, as you can see, diarrhea, and dementia. Finally leading to the 4th D, which is death. So obviously very simple, and I have told you the demarcation also. The distinguishing feature in this image is, you can see there is formation of castle necklace. Whatever area is exposed, it will be pigmented, or there will be excessive, dryness type of skin. Other images I'll show you here. This is again representing this castle. And this is actually named after this Gasper castle. As you can see, this picture is again very clear. Is it clear? Then other options are, if you talk about biotin, biotin is B7. Alopecia is the disorder which is caused by this. If you talk about riboflavin, B2, chlosis is there, glossitis is there due to this. And ascorbic acid, simple vitamin C, deficiency leads to scurvy. So this is again a very simple question which was asked in the exam, and I hope most of the students must have got this answer correct. So again, answer is B. The other name of niacin is nicotinic acid, or it is B3. Then nitrogenous waste is excreted from the body in the form of. Now, this is again a very, very simple question. Let's see what you answered in this. The options were given by as glutamine, urea, uric acid, and nitric oxide. See, if we talk about nitrogen, this nitrogen, the source of nitrogen is actually ammonia in our body, or the amino group, or the source of this amino group is a particular amino acid. So these amino acids undergo a process of degradation. Yes, Mr. Khan, your answer is absolutely correct. The answer is B. Correct. But I know there must be some confusion among the students while attempting the paper. Why can't it be glutamine? Because glutamine is the transport form of this toxic ammonia. Isn't it? This must be a confusion, a doubt must have come in some of the students. Okay, so here simple amino acid will undergo degradation, or this is the, if we compare like what we eat in our diet, we eat carbohydrates, we eat fats, we eat proteins. But out of these three, only the proteins are there who have this amino group in their structure. And they are the only macromolecules which help in disposing of this toxic ammonia in the form of urea. Right? And this is a very, very important cycle which occurs in the liver. It never occurs in the brain. It never occurs in the kidney. The reason is because brain lacks this ornithine transcarbamylase enzyme and kidney lacks the arginase enzyme. If we talk about glutamine, first of all, let me mark the answer here. The answer would be B. So there is no point of confusion. Glutamine, if we say, so glutamine is the simple transportable form. transportable form of this. Because ammonia cannot be left as such in the body, it requires, a carrier. Because otherwise, if it is left free as such, it is going to aminate different acids, or it is going to obviously disturb the acid-base balance. So glutamine is the transportable form which is synthesized from glutamic acid. Right, so it is not the answer, because the question simply says is excreted from the body in the form of, so it has to be urea cycle or urea. And if we talk about the uric acid is excreted, but it is a degradative product of purines. And you know one very interesting thing, in non-primates, this uric acid is degraded to form allantoin by the enzyme uricase. But this enzyme is absent in human beings. Right, and if you look at the fourth option, it is nitric oxide. Nitric oxide is not excreted. In fact, nitric oxide is produced. It is a vasodilator. So the answer is very clear, very simple. It is urea. And urea cycle, the rate limiting enzyme of urea cycle is CPS1. And this occurs in liver in two fractions. One is the mitochondria, other is the cytoplasm. Then we move over to the next question. Which of the following is not a micronutrient? Now see, this is again a very important question and a very simple one. For this, obviously, you have to remember the chart. First of all, you should know nutrients. All of you know nutrient, which is obviously required for the normal functioning of our body. So nutrients, as per their dietary requirement, as per their function, they are macronutrients and they are micronutrients. Now macro means more, micro means less. So obviously, if their requirement is more, they will be macro, and if the requirement is less, they will be micro. Now always remember, even if they are required in less proportion, they are equally important. Isn't it? There are so many micro minerals which are very, very important for our body. So here the question is, yes, very good, Mr. Khan. The answer is C. It is actually a macro mineral. Very good. I am getting the response only from one student in the class, that is Mr. Khan. And he has answered all the questions correctly. So, see, whenever there is a live session, always remember, the more you interact with the teacher, the more knowledge you will gain. That is the, utility, that is the advantage of conducting the lessons live. And that is the benefit of online teaching also. So try to, extract maximum out of the teacher. Because once the lecture is recorded, it will be uploaded, then the teacher won't be available to answer your queries. And always when you know that a particular subject is being discussed, so just be alert for that particular span of time so that you can come up with your problems. Right? And teachers always like those students who actually ask questions in the class. Since this is a recall session, whatever you have studied, whatever queries you had in your mind, that if C is the option, why not A? Why not B? Why not D? So these are the things which has to be asked in this, right? So this is simple. Which of the following is not a micronutrient? So we will see. This is a very good, slide in which you can see these are the macro minerals. These are the micro minerals. The question is simple. Calcium. Calcium is a macro one, right? And if we look at micro minerals, these are the ones, and out of these, every mineral has a very important role to play. Whether it is iron, whether it is zinc, copper, iodine, fluoride, manganese, and selenium. This selenium is a very important antioxidant. So all the questions, all the minerals, whether they are macro, whether they are micro, they play a very, very important role in our body. So the answer to this would be simple calcium, which is a macro mineral. Zinc, copper, and iodine, they are all micro minerals. Right? So I hope whatever questions we could gather from the students, we have tried to present them with full explanation and with each and every option. If this is the option, why not? That is the option. So I hope the questions of biochemistry which I have discussed with you, they were clear to all of you, right? So thank you for your attention. Thanks for listening to all the questions, and I hope most of the students should crack this exam this year. So wishing you all the best, guys. I hope there is no other question. Let me see, is there any query? So you can ask me. Okay, so I'll wind up the session here only. Okay, fine. No more questions. Chalo, take care then. Wishing you all the best. Thank you, Mr. Khan. You like the explanation. Again, I am repeating this. If you guys have any other query, don't hesitate to ask me. Right now, I am on, I am live. So you can ask me. Chalo, let's wind up the session. Take care. Good night. Bye-bye.